Good morning, everybody. I'm Manel Cascallo, the General Director of Theriva Biologics SL, the European subsidiary of Theriva Biologics Inc. And I will take the opportunity just to explain a bit the last updates of our company that has been ongoing in the last month since our last presentation, with a special focus on the data that we have presented at ESMO very recently in Madrid last week. As you remember, Theriva Biologics is a company with an extended pipeline comprising oncolytic viruses and development in different clinical indications: pancreatic cancer, also in retinoblastoma, in head and neck in combination with immune checkpoint inhibitors, but also in a number of different solid tumors, including ovarian, brain, also in collaboration with other investigator-initiated studies in lead sites around the world, including Leeds University and also University of Pennsylvania. The company has also a legacy programs on SIN4 and SIN20 for the prevention of graft versus host disease, and also for the other indications with SIN20. Probably you are perfectly aware of the status of our program in pancreatic cancer, where we are conducting a phase 2 trial, a quite differentiated trial, where we are testing in first-line metastatic pancreatic cancer patients the combination of our lead product, BCN-01, with the standard of care. That it's an open-label, randomized, and multicentric trial that it's being conducted right now in Spain and in US, with an active recruitment right now, where our product BCN-01 who has demonstrated a quite interesting profile in terms of anti-tumor activity and also in safety. It's being tested in a more extended cohort of patients to really confirm the survival and the patients, in order to see if we are able to extend the survival with respect to the standard of care that is administered in the most sites in US and Europe, that is basically a mix of gemcitabine and Abraxane. This trial, it's ongoing, but basically today, what I want to do is basically focus my speech in the new data that we have presented very recently with respect to the efficacy of the trial combining BCN-01 plus immune checkpoint inhibitors. As you remember, BCN-01, it's an oncolytic virus that it's differentiates with other products, basically because it replicates very efficiently, but can be systemically delivered, and moreover express a tumor-degrading protein called PH20 that helps the spreading of the virus into the tumor mass, but also helps the entry of different therapeutic entities, including therapeutic antibodies, but also the immune system. Based on that properties, we designed a clinical trial basically for patients that has been treated with immune checkpoint inhibitors, but failed to the action of immune checkpoint inhibitors, basically because progressed or has never responded to that, with the idea that after the treatment with BCN-01, if we retreat with immune checkpoint inhibitors, the different mechanisms of action of BCN-01 could enhance the therapeutic activity of these immune checkpoint inhibitors. In this trial, we combined BCN-01 plus an anti-PD-L1 antibody called durvalumab, and developed by AstraZeneca, in two different regimens. In one regimen, we tested the virus plus durvalumab concomitantly, it's the arm 1, whereas in arm 2, we tested a sequential administration of first the product and our product BCN-01, and later, 15 days later, we started dosing durvalumab. The idea is that it's in patients with head and neck refractory to immune checkpoint inhibitors, and the idea was to follow the survival of those patients also with other therapeutic indicators, as could be progression-free survival and response rates. One of the beauties of this trial is that in that specific indication, we can get serial biopsies of patients. So from one single patient, we obtain not one single biopsy, but 3 biopsies before treatment, screening biopsy, day 8 after starting the treatment with BCN-01, and day 28 after administration of our product, which provides us an interesting insight of what's the mechanism undergoing on the tumors in order to understand later what's the efficacy data that we obtain. That's the demographic of this study. where we have analyzed different patients in different with different tumors in different anatomical sites. But the interesting thing here, probably I want not to extend in the different technicalities of this trial. In order to understand better the efficacy data, it's important to remember that the safety has already been published for this trial. In fact, we have reproduced basically the safety profile that we have observed in our previous pancreatic trial and other trials that we are conducting. So basically flu-like symptomatology with a very transient elevation of transaminases that is not very relevant, and it's basically asymptomatic and spontaneously resolving, so nothing relevant. And when we analyze it in terms of efficacy, what we have seen is that in these three cohorts of patients, the patients treated in the concomitant arm and in the two arms with the sequential regimen, with the low dose and with the high dose, what we have seen is that the survival of patients, it's quite enlarged progressively in each of these groups. So in the concomitant arm, we have seen a survival of 10 months. In the sequential arm, we have seen a survival of 15 months, whereas in the sequential arm, we have seen a survival longer than 17 months, which is quite relevant in that population of patients that are polytreated, that has experienced several lines of treatment, and they are basically in the last lane of treatment. So what it was the expected survival of these patients before going into the trial is basically in the range between 4 to 6 months. So the data that we have obtained in this trial, it's encouraging. Also, probably it's a reduced population of patients. Each group contained only 6 patients, but are relevant data in terms of it looks like the survival it's enlarged. But the interesting thing is that these patients has also demonstrated a high response rate for all the treatments that they receive after our trial. Because as the life of patients has been extended, that allowed the patients to receive other lines of treatment that otherwise they should be only in palliative cure. And this line of treatment, surprisingly, has resulted much more efficacious than normally expected. In fact, we have seen in the sequential arm several patients with a complete response with the chemotherapy, the standard chemotherapy that patient received after our trial, which also quite significant. Finally, as I said at the beginning, we have collected serial biopsies of the tumor, and that has allowed us to analyze what's going on into the inner part of the tumor. And what we have seen is that there's some significant immunological changes in the tumor in a way that the tumors are much more infiltrated by CD8 lymphocytes, so cytotoxic lymphocytes, but also especially that increase the number of PD-L1 positive lymphocytes, which is a marker of the responsiveness to the immune checkpoint inhibitor. And in fact, when we have analyzed it in a comparative analysis, if there's any correlation between the survival of each patient and the increase in the PD-L1 staining, we see a quite clear statistical correlation for those patients with longer survival with higher staining for PD-L1. So we are encouraged, and we are right now just exploring how to extend the development of this program in BCN01 in head and neck. But these data really support the different mechanisms that we have observed also in our pancreatic phase 1 program, and has been confirmed here, where we have seen also an increase in the survival of patients, as we have seen also in pancreatic cancer, and also several immunological changes in the tumor that justify why this survival is enhanced. So thank you very much for your time, and I hope that that update is useful for you in your consideration. Thank you very much.